Thank you for joining us this afternoon, everyone in the room and online. My name's Karen Jacobs. I'm happy to be able to welcome you here this afternoon to what is the first in what will be a series of microscopy seminars hosted by the IDM Imaging Platform and the New Imaging Center as part of the Africa Microscopy Initiative. So we're thrilled to be able to tell you about our microscopes and to hear in more detail about specifically one of our systems today. Because this is the first session in this series, I am going to start with hopefully I can keep it to 5-minute overview of the Africa Microscopy Initiative and what microscopes we have very briefly before we invite Dr. Munganasap to tell us more about our slide scanning system. So I'm just going to find the right screen and share screen quickly. There we go. Right. So the exciting thing about the Africa Microscopy Initiative is that our imaging center is a no-cost open access facility hosted here at the IDM and the University of Cape Town, but accessible to anyone in Africa. So it's really a resource for the entire continent. And the Africa Microscopy Initiative, or AMI as we like to call it, is a multi-pronged approach to building capacity and access in Africa, which covers an open access imaging center, a program for instrument placement across the continent where microscopes are needed, as well as a number of training programs. For those of you who may have heard about AMI from the big launch we had in October, you would have heard more about the instrument placement programs and the training programs. I'm not going to talk about them today. I'm really just going to tell you about the imaging center because that is the flagship component of this entire initiative, and I think it's the most exciting part. So the Open Access Imaging Center, we also call it a project incubator. The key take-home message is that this is really free to use for any scientists from doing non-profit research in Africa. I will go more into that. So access for anyone, and I'll touch on this again, whether you're coming from down the corridor or across the continent, will be through a peer-reviewed application process. And what we're excited about is that with the systems we have here, we can incubate high potential projects to give them a springboard to other facilities across the world with more advanced systems than what we have. So I'm going to start with hopefully trying to get you interested in why we are focusing on microscopy. And if you think about the sort of our regular toolbox for life sciences, it's actually incredible what we can do. If you think about sort of biochemical assays, ranging from good old Western blots, plate readers, flow cytometry, the incredible technology available under the umbrella of molecular biology, as well as what is now sort of the umbrella of omics. All of these are incredibly powerful and complementary technologies that tell us a lot about the individual components and players that we're interested in, the processes and phenomena that we're studying in our biology. But none of them can truly tell us the mechanism without us getting additional information about the spatial organization and how these components interact in space and time. And that's what microscopy brings as a complementary key component to our research. And I think it's going to be critical for what we do on the continent. So for example, good old-fashioned histology, which you're going to hear more about today, already gives us the spatial context, whether we're interested in a particular structure, cell type or molecule within the tissue. And with fluorescence microscopy and our ability to very specifically label individual chemical environments or molecules, we have that molecular specificity directly into our samples. And of course, the important thing is time as well. And with modern, especially fluorescent microscopy, we can do incredible investigations with live cell microscopy. So this is not a technical, as I said, I'm going to try to keep this to 5 minutes. I've probably used up 3 already. This is not a technical talk. I'm going to do a whirlwind tour of the microscopes that we have accessible to everyone within the facility, and then we're going to hear more about the tissue facts. But you can see we're kind of covering everything, and I'll cover through. If you think about the wide field microscope, this allows us to look from fluorescently labeled tissue sections down to single cell monolayers. We're not limited to cells. Obviously, we can use them for microbiology. This is antibody labeled BCG, as well as up to small organisms. And if we're interested in larger samples, we would be switching, and again, as I mentioned, live cell biology, with the importance of bringing that time component and being able to look at the dynamics of cells, of components, as well as the power that quantification can now bring into that, where you can actually extract key parameters about sort of cell movement, directionality, and other questions that you might be interested in. And if you want thicker samples, of course, your next step would be to confocal microscopy. And this is an image of serial sections through a fly, the eye of a fly, optical sections, not physical sections. So you can really go very deep into a structure or a tissue. And you don't have to have large samples. If you're interested in your molecules within axial space, in Z space or 3D space within your sample, the colors in this image are the axial positioning of the cytoskeleton within a single cell, for example, that you really get that level of information. And again, it's compatible with live cell imaging. So here you will see a red cell appear in a moment. And that is actually a metastatic cancer cell that it's tucked itself in between the edges of two other tissue cells. And that's, you'd not be able to see that without the 3D component of that imaging. The amazing thing about modern confocal microscopes is they also have what's called spectral detection, which allows you to distinguish between not just the typical 3 to 4 fluorescent labels within your sample, but an array of up to tens or even hundreds of labels within a single sample, and really extract a large amount of information from them. If you're interested in higher throughput imaging, that's where it gets very exciting. We can start to talk about slide scanning, and I'm going to touch on this very briefly. Our tissue fact slide scanner for high content imaging. And this is if you imagine you had an array of slides where you want to scan an entire slide, but you don't want to manually sit at the microscope doing every single slide yourself. The system will be able to automatically, with parameters you give it, scan through slides. And the incredible power in the tissue facts and tissue gnostics technology is actually the analytics, in being able to then extract a lot of the quantitative parameters that you're interested in and what they call tissue cytometry, that we'll hear about shortly. And maybe you're interested not in many slides, but in many cell populations, chemical perturbations, drug screening that you might be interested in doing. We have a system for high content screening called the Cell Discoverer, which can allow you to run through on a multi-well plate hundreds of experimental conditions with cultured cells. Again, fully automated acquisition, and the power will be in your quantitative readout. The exciting thing about the Cell Discoverer is it's not limited to only cells cultured in a plate, but it can also be used for almost anything you can fit into the microscope, such as an array of small organisms. These are cricket embryos that have been imaged as an array in the Cell Discoverer. And you can sort of study, again, this is compatible with live cell imaging, so growth and development of small organisms within the system. Sometimes you're wanting to go smaller and not bigger, and there is a resolution limit in microscopy. If you think about that bright green spot on the left would be the signal from an individual, or what you would be seeing from a molecule on your fluorescent microscope. There's a bit of a size discrepancy. It's blocked in this display, but that spot on the left is typically about 300 nanometers in diameter. Your one GFP molecule is 2 to 4 nanometers in diameter. So if you think about where you might locate that individual molecule in your spot, it's a bit of a tracking problem. There's also a bit of a working out how many molecules are in that one spot problem. And that is what we call the resolution limit. That without additional imaging controls, sort of implementing additional technology, you don't have the resolution to work out where and how many molecules you have in your sample. We have two what are called super resolution imaging approaches available in the facility to users. I'm not going to go into the technology at all. I'm just going to tell you one is excellent at identifying single molecules. So it's called single molecule localization, and allows you to really almost count molecules within a sample to go from this is a low resolution conventional image, and high resolution of cytoskeleton. And it's not limited to cell biology. This is localization microscopy used in bacteria to look at transcription factors and viral receptor binding on the surface of a cell, or with localization microscopy. We have a second technology which is more compatible with live cell high resolution imaging called structured illumination. And this can be used for actually very high speed, high resolution imaging. This is of the ER network in a cell. And of course, it doesn't have to be live cells. You can do multi-target imaging within a single cell as well. So that's the overview of the microscopes we have available. How do you access them? I told you have to write a project proposal. This is to highlight, if you're coming from outside the university, the other exciting thing about the facility is that access, cost of travel and lodging is also covered by the initiative. But for local researchers, which I'm assuming most of you are going to be local and interested in how you get access to it, it's actually the same. We just don't worry about your travel and accommodation. You start with planning your experiment, and we encourage you to come and chat to myself and Mike about what it is that you want to do, how you can do it, what quantitative parameters you might be wanting to extract, and what system would be best suited for it. And when calls for proposals go out, you can submit a proposal. It's not a very long one. Our focus on selection is on feasibility, suitability of the system, and what you're going to be doing with that data afterwards. And then we will schedule the time for you to access the systems. When you have access to the systems, you have dedicated access to the systems and support of MicroI to help you run those systems until you are competent to work with them yourself. There is also resources available to help you cover costs of the experiment in terms of labeling, labels and consumables, and there is assistance support available for image analysis afterwards. We also don't operate in isolation. Obviously, the microscopes here are in addition to other microscopes already available within the IDM. So the Microscopy Initiative lab is linked with the IDM facility, as well as the analytical facilities at Stellenbosch. So if there's technology that you need that's not in the AMI lab, obviously it's accessible through local facilities, as well as we mentioned, there's a springboard program that if your project outgrows what's available in the AMI facility, we can link you directly with the Advanced Imaging Center in the US and also the Advanced Bioimaging Unit in Uruguay. We have a sister partnership set up with them. So this is the last slide, just to acknowledge that none of this is possible without very generous funding from the Bill and Melinda Gates Foundation and Chan Zuckerberg, and incredible support from UCT and IDM. Those are the contact details on the left for the Imaging Center overall. Feel free to contact